In this lab, we're going to be determining the number of milligrams of ascorbic acid, also known as vitamin C, in a medium-sized peach. And to do this, we're going to be using the ChemCollective Virtual Lab, and you can find a link to that lab right here. So in this lab, we're going to be taking some potassium iodide, and we're going to be performing a series of titrations. The idea is that we're going to have a sample that contains some potassium iodide, some acid, starch, as well as some ascorbic acid, either as a known solution with a known quantity of ascorbic acid or peach juice, and looking for a color change once we perform the titration. So here is the chemical reactions that we're going to be using. Here we see this is the potassium iodide. This is what's found up here in the burette. The first reaction that's taking place is it's reacting with some potassium iodide and some acid catalyst to produce some iodine. That iodine then goes and reacts with the ascorbic acid to produce this dihydroascorbic acid solution. Now, as long as there is ascorbic acid found within the sample down here in our Erlenmeyer flask, we won't have any excess iodine. It will be constantly being used up. But once we've used up all of the ascorbic acid, there's no more ascorbic acid available to produce this product, and we have excess iodine. That excess iodine can then react with the starch to produce this starch-iodine complex, which eventually turns blue and signals the endpoint of our titration. So to determine the number of milligrams of vitamin C in our peach, we're going to have to do two different parts. One, where we're actually taking the amount of ascorbic acid with a known concentration of vitamin C or ascorbic acid and titrating that. That's where we're using this formula right here. And then we're going to do another series of titrations where we're using peach juice in our Erlenmeyer flask and figuring out what volume of titrant we needed for that to figure out the endpoint. So this is the formula that we're going to be using throughout the lab, and this lab is going to have a series of parts. Part A, we're going to be creating a stock solution of potassium iodide from a solid. So this is the stuff that we're putting up in our burette, and that has to be created in the lab. It's not provided as a solution that we can just add to our burette. We actually have to make it. And you can take a look at the content page about creating stock solutions to figure out how many grams of this potassium iodide you're going to need to create a solution with a known molarity and volume. So check that out. And then in part B, we're going to take those sample solutions of ascorbic acid with that known concentration of vitamin C and performing a titration. So we know that when we do part B, the ascorbic acid solution that we're using has 20 milligrams of vitamin C if we've prepared it correctly. Then we're going to have to figure out in part B how much volume of titrant we need for that color change to take place. Once we have that, we have this component of the formula that we're going to be able to use to later calculate the milligrams of vitamin C in a peach. In part C, what you're going to be doing is you are going to be designing a procedure where you're going to be putting the peach juice and preparing the sample so that it can then be titrated to figure out how much volume of titrant you needed to make a color change take place within your peach solution. That means that you have this component from part C and this component from part B, and we can solve for the milligrams of vitamin C in peach juice. So as a part of this lab, you're going to be working through the virtual lab, going through part A, going through part B, and eventually the end goal is to create a lab report. You can see what's needed in the lab report by taking a look at the very bottom of the page. There's a link to how to write a correct lab report, and these are all the components that you need to include in your lab report, and you can take a look at this content page for information and details on what should be included for each of those sections in a formal lab report. Now, once you've done all of the titrations, you're eventually going to figure out how many milligrams of ascorbic acid or vitamin C are in your medium peach, and you're going to enter that into this little box right here that's found at the bottom of the lab in the ChemCollective site. Now, what you should be doing is you should be doing 3 different trials for every single sample. or sorry, every sample should be titrated 3 times so that you can average the volume of titrant that you're using to figure out the volume of titrant used for your titration. And then you can do your calculation and figure out how many milligrams of ascorbic acid are in that peach. Now, every single peach in the lab is different, so you can't just take a look at something that you find online. You actually have to do the lab to get the right result. And if you do the lab correctly, you will eventually see a page that says, yeah, your answer is correct. The medium peach that you used contained this amount of ascorbic acid, and your answer is correct. As you're working through the lab, you're going to be asked to take screenshots of your work. You're going to have to rename your flasks with your name, as well as what's indicated within the instructions. And those screenshots just let me know that you've actually performed the lab yourself and that you're not taking information or something from the internet and passing that off as your own work. I want to see that you've actually performed the lab. So that is the formal lab report. It will just be submitted to a D2L Dropbox, and you'll be graded using those skills rubrics found at the bottom of the page. The lab can be a little bit involved, and if you have any questions, make sure you contact me in private discussions.